All right, let's dive right in. Today, we're going on a pretty incredible journey. We're going to see how scientists take this absolute storm of raw DNA data and somehow, almost magically, turn it into a crystal clear map of your genetic code. So, to really get a sense of the challenge, I want you to imagine something. Imagine your entire genetic code is a massive encyclopedia. Not one book, but a whole set of them, filled with 3 billion letters. Now, your job is to read through that entire thing and find every single spelling mistake, every single typo that makes your copy unique. That is the exact scale of the problem we're talking about. And this giant encyclopedia of life? Well, that's your human genome. The whole point of the process we're about to walk through is to find those specific typos. Now, in genetics, they're called variations. And these little differences are the key to, well, everything. They explain our hair color, our height, and even our risk for certain diseases. So, where does a mission like this even begin? Well, before you can find any typos, you need the original text, right? In our case, that means getting our hands on the raw DNA data that makes up that genetic blueprint. And that raw material comes from this incredible technology called next-generation sequencing, or NGS for short. You can think of it as a super powerful DNA reading machine. Now, sometimes scientists use it to scan your whole genome. Yep, all 3 billion letters. Or to make things a little faster and cheaper, they might just look at the whole exome, which is the 2% of your DNA that actually contains the instructions for making proteins. But either way, NGS is the machine that spits out all that initial raw data. And this is where the hero of our story comes in, the FastQ file. And let me tell you, it's not a very glamorous hero. At first. The sequencing machine can only read DNA in short, little bursts. So what it does is it basically takes that 3 billion letter encyclopedia, puts it through a shredder, and gives you millions of tiny, jumbled sentence fragments all thrown into one big digital pile. That pile is the FastQ file. It's messy, it's chaotic, and it's full of errors, what scientists call noise, from the machine itself. So what's the treasure we're actually hunting for in this jumbled mess? We're looking for something called germline variants. These are the typos we were born with, the ones we got from our parents. They're in every single cell in our body, and these are the specific differences we're trying to find. Now, obviously, you can't sort through millions of shredded pieces of paper by hand. No way. To turn this absolute chaos into order, scientists, specifically these digital wizards called bioinformaticians, need a really powerful and specialized digital toolkit. And this table here, this is basically our itinerary for the entire journey. These are the actual software tools that are going to transform our messy data one step at a time. You've got tools like FastQC to check if the data is any good, BWA-MEM, which does the really heavy lifting of putting the puzzle together, and later on, this master detective called GATK HaplotypeCaller that actually spots the typos. Okay, toolkit's ready. It's time to put our data through what you could call a digital gauntlet. This is where we take those millions of jumbled fragments and finally start assembling our genetic puzzle. And this journey really has 4 main stages. First, we gotta clean up the raw data. Then we assemble it all against a master copy. After that, we refine the picture, make it sharper. And finally, the really exciting part, we go looking for those unique variations. So phase 1 is all about quality control. You can't build a story if half the pages are smudged and unreadable, right? So tools like FastQC and FastP go through each and every fragment, check its quality, and digitally trim away all the low-quality bits. It's literally like erasing all the smudges from the edges of the page. Okay, now this next step is pure computational magic. A tool called BWA-MEM takes our millions of clean fragments and compares them to a standard, perfect human genome. Think of it as the master copy of the encyclopedia. Incredibly, it figures out exactly where each tiny fragment belongs, assembling our puzzle piece by piece on a massive scale. But how do we know if this is even working? I mean, that sounds pretty intense. Well, here's the key metric they look for. A really good, successful alignment means that over 95% of our DNA fragments have found their correct home on that reference map. That's how you know you've got a rock-solid foundation to build on. Now, before we get to the big finale, we have to do a little bit more cleaning up. You see, the lab process itself can sometimes create artificial duplicates of the same DNA fragment, kind of like accidental photocopies. A tool called Picard is amazing at sniffing these out and removing them, making sure we don't get tricked by a typo that isn't really there. Okay, the data is clean, it's assembled, it's refined. The puzzle is basically complete. And now for the moment we've all been waiting for, it's time to actually spot the differences. This really is the climax of the whole thing. A super powerful program from that GATK toolkit meticulously scans our assembled genome, comparing it letter for letter against that master reference copy. And whenever it finds a difference, it flags it. This is the big aha moment. The software is finally circling the typos in our book. But there's a catch. Here's the crucial final question. The process isn't perfect. Sometimes the sequencing machine itself just hiccups and makes an error, creating something that looks like a typo but is really just a digital smudge. So how in the world do you tell a real genetic difference from a simple machine error? And this slide right here shows you exactly how they do it. It's called variant filtering. Scientists use really strict criteria to build confidence. A real, high-confidence typo is going to have strong evidence. You'll see it over and over again in all the different DNA fragments that cover that one spot. An error, on the other hand, it's going to be a weak, noisy signal, maybe only showing up once or twice. It's the difference between a real typo and a machine smudge. So, after passing this final, rigorous test, our data's long journey is finally complete. From that initial storm of this chaotic information, we have created something incredibly organized, incredibly precise, and incredibly valuable, a personalized genetic map. And that map has a name. It's called the Variant Call Format File, or, for short, the VCF file. This is the treasure at the end of our entire computational quest. You know, what's so amazing about the VCF file is its efficiency. It's not trying to rewrite your entire 3 billion letter genome. That would be crazy. Instead, it's just a simple, concise list of the coordinates, the exact spots where your genetic encyclopedia is different from the reference copy. It's a map that leads directly to everything that makes you unique. And each entry on this map is just so elegantly simple. It basically just says, okay, on this chromosome at this exact spot, the reference book has a G, but you have an A instead. That's it. A precise list of your genetic individuality. So why do we do all of this? Why does any of this matter? Because this final map is what unlocks the future of medicine. It's what allows researchers to pinpoint the exact genes linked to inherited diseases. It helps doctors understand why you might respond differently to a medication than someone else. It is the absolute foundation of personalized medicine. So, if there's one thing to take away, it's this incredible transformation. We start with this chaotic storm of millions of disconnected, messy data points. And through this powerful bioinformatics journey, we end up with a clear, precise map of what makes a person genetically unique. And with that map in hand, well, that's when the real search for answers about our health and our history can truly begin.